What if we could fight cancer not from the outside in, but from the inside out? What if we could actually turn a tumor's greatest defenses against itself? Well, that's exactly what we're talking about today. We're diving into this incredible, revolutionary science of using genetically engineered bacteria, basically microscopic assassins, to hunt down and destroy solid tumors.
I know, it sounds like something straight out of science fiction, right? But believe it or not, this is happening right now in labs all over the world. Scientists are literally programming these tiny, single-celled organisms to become living, autonomous medicines. So how on earth does this even work? Well, to get it, we first have to understand the enemy.
So for decades, solid tumors have been this massive challenge for doctors. They build a literal fortress around themselves. It's this complex ecosystem that is just actively hostile to pretty much every treatment we can throw at it. And right at the heart of this fortress is something called the necrotic core. See, as a tumor grows, it grows so fast that it basically outruns its own blood supply. So the center gets starved of oxygen, and it creates this dead zone of dying cells. And this core? It acts as a perfect physical and physiological shield. And this is the problem, right? You've got this tangled mess of blood vessels and intense internal pressure. It all acts like a wall. It stops chemotherapy drugs from ever reaching the tumor's core. And at the same time, that low oxygen environment inside, it's a condition called hypoxia. It pretty much disables our body's own immune cells, our own natural defenders. It's a perfect storm of resistance. So how do you get past an impenetrable fortress? Well, you don't try to break down the walls. You send in an agent that actually thrives in those exact conditions.
And this is where our story takes a really interesting turn. The hero in this new approach? It isn't some powerful new chemical. No, it's one of the oldest and simplest life forms on Earth, bacteria.
Now, the idea itself isn't totally brand new. Way back, over a century ago, a surgeon named William Coley noticed something strange. Some of his cancer patients who got these severe bacterial infections actually went into remission. His Coley's toxins were a really crude first attempt. The huge difference today is precision. We've gone from using a blunt instrument to programming a microscopic smart bomb.
And what's amazing is that scientists now have this whole toolkit of bacteria to choose from. They can pick just the right one for the job. Take Clostridium, for example. These guys are what you call obligate anaerobes, which basically means oxygen is poison to them. So where do they go? They will only grow in that zero-oxygen necrotic core of a tumor where they literally just eat the dead tissue. Then you've got others, like Salmonella. They're super mobile and can be used like these tiny little microscopic trucks to deliver cancer-fighting drugs deep inside the tumor.
Okay, but you're probably thinking, how do you control them, right? How do you give a simple little bacterium a complex mission? And maybe more importantly, how do you install safety switches? Well, the answer is just incredible. It's in this field called synthetic biology. There's a great way to think about this. It's like building an electrical circuit, but instead of using wires, you're using pieces of DNA. Scientists are actually building genetic circuits. These are like logic gates made of DNA that can sense the environment and then trigger a really specific action, almost like a tiny computer program. Let's look at one of the cleverest systems they've come up with.
It's all based on a natural process called quorum sensing. And you can think of it just like a biological roll call, or maybe like shouting in a room. Each little bacterium releases a chemical signal, and as the population grows in one spot, the concentration of that signal, the shouting, gets louder and louder.
And here's where that roll call becomes this absolutely critical safety switch. So first, the bacteria find and colonize the tumor's zero-oxygen core. As they multiply, those chemical signals start to build up, right? Just like the volume in a crowded room. Once that signal hits a certain level, a critical threshold, it flips a genetic switch inside the bacteria. And this switch turns on a brand new ability, oxygen resistance. Now this is the crucial part. It means they can now survive at the tumor's edge, but, and this is the key, only after they've already built a big safe colony deep inside. This is what prevents them from just growing anywhere else in the body. It is an absolutely brilliant piece of biological engineering. By the way, if you're finding this look into the future of medicine as fascinating as I am, hey, take a quick second to like this and subscribe for more deep dives into this kind of cutting-edge science. It really helps us out.
Okay, so we've got our bacteria. They can find a tumor and they can safely set up camp inside it. But now, how does this actually become a therapy? How do they deliver that final blow?
Well, one of the most advanced techniques is something called a synchronized lysis circuit.
Okay, so the bacteria are programmed to be these tiny, self-contained drug factories. But here's the cool part. Instead of just slowly leaking out the medicine, they wait. They wait until their population hits that critical mass inside the tumor, and then, bam! The genetic circuit triggers them to all burst open, or lyse is the scientific term, at the exact same time. This releases this massive, super concentrated wave of therapy right where it's needed most. And then a few bacteria survive to recolonize and repeat the whole cycle, creating these therapeutic pulses from deep inside the tumor.
And listen, this isn't just theory anymore. This isn't just in a petri dish. This technology is actually moving into human trials. One of the leading candidates right now is a therapeutic called ACTM-838, and it's being studied in patients with advanced solid tumors as we speak. And just look at the potential range of this thing, because this whole approach targets a fundamental feature of pretty much all solid tumors, that necrotic, low-oxygen core. It has the potential to be a powerful new weapon against so many different types of cancer, especially the ones that have been really, really hard to treat.
So let's step back. What does this all mean for the bigger picture? Honestly, it represents a fundamental, game-changing shift in how we even think about attacking cancer.
Yeah, this really sums it all up, doesn't it? For basically a century, we've been trying to blast our way into the tumor's fortress from the outside. Bacterial therapy? It's the ultimate Trojan horse. It doesn't fight the tumor's environment. It uses it as a sanctuary, as a hiding place. This inside-out attack is so highly targeted, it localizes the therapy right where it needs to be, and that could dramatically reduce the awful systemic side effects you see with traditional chemotherapy.
Of course, a therapy this revolutionary needs new rules, right? The FDA actually classifies these engineered organisms as biologics. That's the same category as things like vaccines and gene therapies. It's a totally new frontier. And the good news is that regulatory bodies are actively working on creating flexible new pathways to help speed up the development of these incredibly promising living medicines.
And really, that is the powerful, central idea behind this entire field. It's about using our understanding of a tumor's unique biology to transform its most formidable defense into the very key to its own destruction. We're sending in microscopic allies to dismantle the fortress from the inside out.